So my project is on osmosis in a leaf. And I wanted to know, well, how do different solutions affect the cells in a leaf? So cells in leaves are affected by what's called osmosis. And osmosis is a type of diffusion where water travels across a semi-permeable membrane, such as cell or animal, plant cells or animal cells, and it travels from the least, from the most concentration of solute to the least. Okay, and so there are several types of solutions. In an isotonic solution, that means that the solution has the same amount of solute on the outside of the cell as it is on the inside of the cell. In a hypotonic solution, there is less solute in the solution on the outside of the cell than there is on the inside of the cell. And so then water will travel from the inside of the cell to the outside to achieve equilibrium. In a hypertonic solution, there is more solute on the outside of the cell than there is on the inside of the cell. And so water will travel out of the cell to obtain equilibrium. And so our hypothesis is that when I place the leaves in solution, when it's placed in the water solution, that there will be no change in the leaf. When I place it in solution 1, which will have salt dissolved in the water, and solution 2, which will have sugars dissolved in the water, that there will be osmosis will occur where the water will come outside of the cell and the leaves will shrivel. Solution 3, which contains vinegar, there will not be any change in the leaf. So my variables in the experiment are, in my control variables, is that all the leaves were from the same tree. They were placed in the same place with the same amount of light and temperature. And they were all placed in the same type of container. My independent variables of the experiment are the solutions. One container contained a cup of water. Then solution 1 contained a cup of water with salt dissolved into it. Solution 2 contained a cup of water with sugar dissolved in it. And solution 3 was a cup of vinegar. So these are the materials that I need for my project. I need 3 cups of water, storage container, a tablespoon of salt, leaves, preferably 5, tablespoon of sugar, measuring cups, measuring spoons and a measuring cup, a cup of vinegar, my data sheet, and labels. Okay, so this is how I'm going to do my experiment to see how leaves react in certain solutions. So I've gotten my containers and I have them labeled with the labels: control, solution 1, solution 2, and solution 3. So what I'm going to do is I'm going to pour 1 cup of water into the control. Then I'm going to pour a cup of water into solution 1 and a cup of water into solution 2. And then into the bin labeled solution 3, I'm going to pour my vinegar. Okay, so I am going to take my salt and I'm going to pour it into the bin that is labeled solution 1. And I'm going to stir it up, make sure that it dissolves. as much as possible. Okay. Okay, so that's pretty much dissolved as much as possible. All right. And then I'm going to take the sugar, tablespoon of sugar, and I'm going to pour it into solution 2. Okay. And I'm going to stir this up as much as possible. Okay. Okay, so now I have my control, my 2 hypertonic solutions, and my vinegar. Okay, and so now what I'm going to do is I'm going to place a leaf in each one and make sure that it's covered. First, I'm going to note that how each leaf is. So this is the first leaf that's going in the water, and it's firm and it's green, as you can see, and it's going in the control solution. This is my second leaf. Okay. And this is also firm, and it's going into my second solution, which has salt in it. Okay, this is my third leaf. My third leaf, and it's firm, in good condition, and I'm going to put it in my sugar solution. And this is my fourth leaf, which is firm, and it's going into my vinegar solution. Okay, and now I will set the timer, which is on my phone, for 30 minutes. So this is after overnight, the end result of our experiment. This is our control leaf, and as you can see, it has stayed pretty much the same. This is the leaf that has been placed in water, and it is still rigid. As you can see, even more so than it was before. This is our leaf placed in salt water, and this has become more flexible and soft. Okay. This is our leaf placed in solution 2, which is our sugar. Okay, and as you can see, this turned out to be very flexible and soft also, where it has lost some of its rigidity. And finally, this is our leaf that was in vinegar. Okay. Okay, and that has also remained pretty much flexible and soft also. So the results of my experiment showed that that at the end, where the leaves were placed overnight, that the leaf that was placed in the water actually became more firm, And the two leaves that were in solution 1 and solution 2 were less firm but not withered. And the leaf that was placed in the vinegar had the most effect, where the leaf was the most pliable and more flexible. I found that my hypothesis was not supported, in that the water, which actually ended up being the hypotonic solution, caused the leaf to become more rigid. And that is because the water traveled inside the leaf cells. Solution 1 and solution 2, even though they became less flexible, they did not wither. And this may have been because the cell walls were were stronger than expected, and it maintained its structure. In solution 3, which was the vinegar, my hypothesis was not supported because that actually had the most change. And so further investigation will show whether the leaf was the least flexible because the vinegar broke down the cell walls, or whether osmosis actually occurred. So whether you have a green thumb or a black thumb, as I do, plants communicate their needs to us by the appearance of their leaves. When they're in need of water, then they are drooping. That means that they need to be watered, and water is the best solution because it causes osmosis, water to go into the cell, which is what is needed. Thank you.